Good morning, my dear students. In the last class, we had discussed about the glycolysis. The end product of the glycolysis is pyruvate. The pyruvate enters the citric acid cycle to acetyl-CoA. So today, let us discuss about the citric acid cycle. Citric acid cycle, also known as TCA cycle, tricarboxylic acid cycle, or the Krebs cycle, is a series of chemical reactions used by all aerobic organisms to release and store energy through the oxidation of acetyl-CoA derived from carbohydrate, fat, and protein into adenosine triphosphate and carbon dioxide. All the enzymes catalyzing various reactions in the pathway are localized in mitochondria. The TCA cycle occurs in the matrix of mitochondria in eukaryote. In prokaryote, it takes place in cytosol. The cycle includes 8 major steps. Before the starting of the citric acid cycle, first pyruvate is converted into acetyl-CoA. Pyruvate is oxidatively decarboxylated to acetyl-CoA. This reaction is catalyzed by a group of enzymes called pyruvate dehydrogenase complex. This is the reaction of the for the conversion of pyruvate to acetyl-CoA. Pyruvate is oxidatively decarboxylated to acetyl-CoA in the presence of NAD and coenzyme A. The product of this reaction, that is the acetyl-CoA, is the starting point of the citric acid cycles. Now, let us start the process of citric acid cycle. In the first step, the citric acid, first step of the citric acid cycle, acetyl-CoA combined with oxaloacetate to form citrate. A molecule of water is consumed and CoA is released. This reaction is catalyzed by citric synthesis. It is an irreversible reaction. In the step 2, citrate is converted into isocitrate by the enzymes aconitase in 2 steps. In the first step, citrate is dehydrated to cis-aconitate, which is then rehydrated to form isocitrate. This reaction is inhibited in the presence of fluoroacetate. In third step, this isocitrate is then oxidized to alpha ketoglutarate, which results in the release of carbon dioxide. 1-NADH molecule is also formed in this step. The enzyme responsible for catalyzing this step is isocitrate dehydrogenase. This is a rate limiting step, as isocitrate dehydrogenase is an allosterically controlled enzyme. In the fourth step, alpha ketoglutarate is oxidatively decarboxylated to succinate-CoA. The mechanism of this reaction is similar to that of oxidative decarboxylation of pyruvate. This reaction is irreversible and catalyzed by alpha ketoglutarate dehydrogenase in the presence of TPP, lipoic acid, FAD, and NAD enzymes. In the fifth step, succinate-CoA, this succinate-CoA is converted into succinate by the enzyme succinate thiolase. The conversion is accompanied by the formation of a high-energy phosphate bond in the presence of GTP and inorganic phosphate. GTP then transferred to high-energy bond to form ATP. This type of phosphorylation is called substrate-level phosphorylation. In the sixth step, succinate undergoes. The succinate undergo dehydrogenation to form a 4-carbon molecule called fumarate in the presence of succinate dehydrogenase and FAD, which is converted into FADH2. In the 7th step, fumarate is hydrated to form malate in the presence of the enzyme fumarase. Although no coenzyme is required in this reaction, phosphate is believed to have activating the effect. In the last step of this cycle is the conversion of malate to oxaloacetate. It is a dehydrogenation reaction. This reaction is catalyzed by malate dehydrogenase in the presence of NAD+, which is reduced to NADH in the process. Then product of the citric acid cycle. Each cycle produces 2 molecules of carbon dioxide, 3 molecules of NADH, 3 hydrogen ion, 1 molecule of FADH2, 1 molecule of GTP. As such, each molecule of glucose produces double this. That is 4 carbon dioxide, 6 NADH, 6 hydrogen ion, 2 FADH, and 2 GDP. These enzymes are reoxidized through the respiratory chain enzyme of mitochondria, yielding 3 ATP molecule per molecule of NADH and 2 ATP molecule per FADH molecule. And then let us discuss about the energetic of the citric acid cycle. The total number of ATP formed during the oxidation of pyruvate through citric acid cycle is, in the conversion of isocitrate to oxaloacetate, 1 NADH is produced, so 3 molecule of ATP are produced. In the conversion of alpha catechol to succinate-CoA, 3 molecule of ATP are produced. In the conversion of succinate-CoA to succinate through 1 molecule of ATP is produced. In the conversion of succinate to fumarate, 2 molecule of ATP is produced. In conversion of malate to oxaloacetate, 3 molecule of ATP is produced. So total number of ATP produced per molecule of acetate or acetyl-CoA is 12. So as 1 molecule of glucose give 2 molecule of pyruvate, that is 2 mole of acetyl-CoA, total number of ATP formed in citric acid cycle should be 12 into 2 is equal to 24. Thank you, my dear student.